So, today we're diving into a study that shows that switching off one crucial protein, ferritin light chain 1, or FTL1, appears to actually reverse brain aging in mice.
That's right. Wait, are we saying that by simply dialing down this protein, the brains of old mice seem to get younger?
Exactly, it's pretty remarkable. The study, led by researchers at UCSF, found that in the hippocampus, that's the area key for memory and learning, old mice had a lot more FTL1, while young mice had less.
Yep, I got that. And the experiment, correct me if I'm wrong, involved using genetic editing to artificially increase FTL1 in young mice and reducing it in the older ones?
That's right, they basically overexpressed FTL1 in young mice, which made them show signs of accelerated aging. Think impaired memory, poor learning skills. And then, by reducing FTL1 levels in old mice, they saw some, it's like, almost miraculous, signs of restored cognitive function.
Absolutely. Wow. So it's not just about stopping aging symptoms, it's almost a reversal of impairments. But, isn't it a bit early to call it a magic bullet for, say, human conditions like Alzheimer's?
You're spot on, and that's a really good challenge to consider. The researchers themselves caution that this has only been demonstrated in mice. There's still a long road before we can apply these findings to humans, especially considering the complexity of conditions like Alzheimer's and
Totally.
Parkinson's.
And the mechanism here, is it all about FTL1 interfering with the normal neuronal connections in the brain?
Yes, exactly. Neurons with high levels of FTL1 showed fewer branching arms, which are crucial for connectivity among nerve cells. It's like it's like having a communication network where the wires or branches are frayed. This protein also seems to interfere with the mitochondria, our cellular
Right, that's
powerhouses,
fascinating.
which is another key factor in aging.
So, in a way, this study connects protein mismanagement with mitochondrial deterioration, both of which are known to influence aging.
Exactly. It suggests that FTL1 isn't just a bystander but might be a driver of brain aging, at least in these mice models. And the hope is that with more research, we might be able to design therapies that target similar pathways in humans.
Yeah, that's a big leap. It sure is. But I'm curious, aren't there potential risks in reducing FTL1, considering that it's normally involved in storing iron in the body?
That's a valid concern. FTL1's normal role is indeed iron storage. Too little of it might create other issues. So, any therapies would need to strike a delicate balance. It's not as simple as flipping a switch. We need to carefully modulate its levels to avoid unintended consequences.
Absolutely. I see. So, the study really opens up a promising new target for potentially combating neurodegenerative diseases, even if it's still preliminary.
Yes, it's an exciting, albeit early step in the right direction. It's like catching a glimpse of a future where we might better maintain or even rejuvenate brain function despite aging.
Right on. Incredible stuff. And while we understand that translating this from mice to humans will be challenging, it's great to see such innovative research pushing boundaries.
Exactly. It's a hopeful time for aging research. Bold ideas are paving the way. And who knows, maybe one day this line of work will lead to breakthroughs in treating neurodegenerative conditions.
Absolutely. Well, that's all we have for this deep dive. Thanks for breaking down the details. It's been really enlightening.
Thank you. It's been a pleasure chatting about this fascinating research. Until next time.